Alright, let's look at protein synthesis. We know that we are multicellular organisms and that our cell's job is to make proteins. And the directions for those proteins are going to be found in our DNA. They're coded as genes. But how do I actually go from a gene to a protein? That's called protein synthesis. And the way your cells do that is it takes your DNA and it reads it. And the one reading it is something called RNA polymerase. So RNA polymerase is just going to sneak in there on one side of DNA, and it's going to start looking at the bases that are along that strand. And it's going to read them along. So imagine this is my DNA strand. Excuse me, sorry I'm wiggling so much. I have my bases all paired up according to DNA base pairing. And my RNA polymerase is just going to run along and read it. But I'm changing the type of nucleic acid I'm dealing with. I'm not making DNA. I'm switching to something called RNA. So some of my base pair rules apply, some of them have to change. Because I'm going from one type of nucleic acid to another type of nucleic acid. And that's why I'm stressing that we're using RNA polymerase. If I want to make more DNA, I use DNA polymerase. If I want to make something different, I'm going to use a different enzyme, in this case RNA polymerase. So as my RNA polymerase is reading this, it's going to base pair according to a different set of rules. So what happens is, if I have a DNA strand and I have an adenine and I want to make it into RNA, the A has to go to U. If my thymine is here, it's going to go to A. And my C is going to go to G, and my G is going to go to C. Everything's almost the same except for this U. There's no thymine in RNA. I'm only using these bases. So if I'm switching my DNA over here to RNA, I gotta use these rules. So this A, we'll just write it underneath, goes to U. My T goes to A. My C goes to G. My G goes to C. My T, A, U, C, G, U. Now, I have copied my gene. I'm going to take this to the ribosome. So we're going to go to the ribosome. So I can take this gene now that I've made into RNA, particularly it's called messenger RNA, and I'm going to convert it to a protein. How I read this is 3 letters long. Each 3 letters is called a code. It's coding for a specific amino acid, and I'm going to use a wheel to look up each codon. So my first codon is AUG. I start in the first circle, because that's my first codon, is U. 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 Then I go to A, so that's my second one. I'm going to look for the A that's over in this quadrant for my U, which is right here. My third letter is a G, so I look for my G, and I actually end up with a stop codon, so it's a bad example. Let's try the third one. CAU. C-A-U. Histidine. That CAU codes for this amino acid. And I would start stringing them together. And I think we got that really clear when we were doing our sentences, but I want to show you like how all this ties back to you and actually creating you. So I'm going to let you use this example, which is called the snorks. So we have these fictitious little characters called snorks, and they have one chromosome with 10 genes, and each gene codes for a different trait. just like your genes code for different traits. Those genes code for amino acids. And so in this amino acid sequence, this is actually protein, a full protein. Okay, imagine these are proteins. These are the amino acids that make up that protein, because remember, proteins are made out of amino acids. Now I can have different versions of a gene. That's why we have blue eyes, brown eyes, blonde hair, red hair. We get different versions of genes that can code for different proteins that are going to result in different traits. So what we're going to do is we're going to figure out what does one of these snorks looks like. So you can do Snicker Snork, Snuffle Snork, you can do Snapple or Snoopy. So let's stick with Snoopy. This space right here is indicating that's gene 1. This one is gene 2. Then we've got gene 3, gene 4, gene 5. This is the 6th gene, 7th gene, 8th gene, 9th gene, and 10th gene. So let's just pick a nice short one. Let's look at gene 9. I have my letters broken up into groups of 3, so I have my codons. I'm going to use my wheel to figure out what is it coding for. Well, my first one was U, U, U. So come over here, I have U, U, U. It makes up phenylene, so I'm just going to put PHE over here. The second one is UCU. UCU, serine. The last one, GGU. GGU, glycine. I don't have to do anything else other than just look this up because they've already gave me the RNA. Now I got to go to gene 9, which is right here. Oh, we're coding for ears. And match. I got phyline, serine, glycine. Phyline, serine, glycine. So I match this one, which means they're going to have pointed, standing up ears. So you're going to do that for each gene, figure out what they're going to look like, and then you're going to draw it out and turn it in. If you get stuck or have any questions, just let me know and I will help you out.